Good morning, everybody. Okay, welcome back to our symposium. Okay, so we are having symposium titled Innovations in Material Biology Interface in Dentistry. And today we have three speakers. So let me introduce our speakers. The first speaker is Associate Professor Vinicius Rosa. So Dr. Rosa is an associate professor at the National University of Singapore, at the Faculty of Dentistry and also Faculty of NUS Center for Advanced 2D Materials. So they are studying the potential of graphene family materials to induce osteogenic differentiation and mineralizations to delayed biofilm formation on titanium. And actually, Dr. Rosa has been receiving different awards from Academy of Dental Materials Student Awards, George Pathan Badger Student Research Award, Ideal Distinguished Scientist Young Investigator Award, and the IDL DMG Stephen Bain Mid-Career Award. So right now, Professor Rosa is the Vice President of the IDL Dental Materials Group, and he is also an Associate Editor for the Journal of Prosthodontics. And our next speaker, the second speaker is Professor Melanie Sordano Damel, who is also the chairman of this symposium. Professor Melanie is a professor in biochemistry, biomolecular, and oral biology at the Faculty of Dentistry, Prasasti University, Indonesia. She is the head of Indonesia Dental Council, Indonesia Medical Council, with a main responsibility in improving continuous dental education, registration of dentists in Indonesia, and overall development of dental profession. She is the past president of our IADL SA division from 2015 to 2017. She has been conducting research mainly on utilizing natural agents in dentistry. So Professor Melanie has received an honorary PhD in oral health science, Faculty of Dentistry, Thammasat University, Bangkok, Thailand. Our last speaker is Professor Po-Chun Chen from the National Taiwan University. So Professor Chan is currently the director of the Graduate Institute of Clinical Dentistry, School of Dentistry at the National Taiwan University. He obtained his PhD at University of Michigan and also specialized in periodontology at National Taiwan University. So Professor Chan is a well-known researcher in the fields of tissue engineering, regenerative medicine, periodontology, and implant dentistry with publications of reputable journals. He is the editor-in-chief for the Journal of Taiwan Academy Periodontology and the Journal of Periodontics and Implant Dentistry. So we thank you all the speakers to speak today. And then now I would like to pass the time to our chairman, Professor Melanie. Professor Melanie, please.
Okay, thank you very much. I just tried to open the mic. Thank you very much, Professor May. I'm very happy and very delightful to have the 35th annual meeting of IADR Southeast Asia Division. I'm so happy that we can meet, although with other participants, not face-to-face, but anyway, hopefully what we give today to you all can bring more to dentistry in the future. Okay, I think we can start the first speakers from Associate Professor, Dr. Professor Rosa, to give the presentation, graphene nanocoating to improve osteogenesis and decrease biofilms on titanium. I think that we can share the video. Thank you.
Hi, my name is Vinicius Rosa. I'm from the Faculty of Dentistry of the National University of Singapore. And today I'm gonna present to you the graphene nanocoating to improve osteogenesis and decrease biofilm on implants. So when we think about dental implants, we often are challenged with these two different scenarios. Either it takes too long to integrate, up to 6 months, according to some reports, or when the integration goes well and the patient start using the implant and living with that, the patient can have this perimplantitis disease that affect up to 20% of the patients. So it's a very prevalent problem. And perimplantitis that can culminate in the loss of the implants. So many people have already tried different solutions to solve this problem on the osseointegration side. For instance, one very traditional example is apatite coatings that can help to induce the bone formation, but they also may delaminate over time because of the thickness, mechanical properties that are not that great. And even the hydroxyapatite coatings have been correlated with higher degrees of marginal bone loss, what is certainly not beneficial to implants. On the infection side, we have a paradox, right? The paradox of antibiotic coatings. Antibiotic coatings are very, very popular strategy that people tried. So in this paradox, you can go for a strategy of slow release of the antibiotic to kill biofilms over time, but that can also lead to antibiotic resistance. Or you can just have a short release of the antibiotics and kill all the bacteria at once, but then you don't have any protection over time. So this is also a solution that it's often incomplete. In our lab, in the last 8 years, we have exploring graphene to improve biomedical implants, namely titanium implants. And what is graphene? Graphene is a single atomic sheet of carbon atoms arranged in a honeycomb structure. And what is that honeycomb structure? Is several aromatic rings arranged side by side, like you can see in this AFM picture. And graphene has some very interesting physical and chemical properties, but we're going to focus on some that seem to be more prominent, more interesting for biomedical applications. It's very thin, strong, and stiff. It's transparent, stretchable. It's completely impermeable, and it also has a very large surface area. So that means you can coat objects without increasing the thickness or the weight of these objects. And our work was inspired from this work from 2011 from Prof. Barbaros, also here from NUS, where they have observed that bone marrow stem cells on graphene had higher mineralization potential regardless of the presence of BMP2. We know that BMP2 can induce a lot of osteogenic differentiation, but here in this panel F, you can see that even without the BMP2, the cells on the graphene present quite a lot of mineral content. So we have checked a few years later by ourselves in a study using periodontal ligament stem cells. And in this work, as you can see, the cells were seated on both graphene films, what we call 2DGP, and graphene scaffolds, a 3DGP. And what we see after 5 days, cells are well, right? So they have attached, they proliferate, they extend on the surface. But when we look at the osteogenic differentiation, then things starts becoming very exciting. So first of all, if you see here, you're going to start seeing some green proteins. That means these are osteocalcin, which is a marker for terminal differentiation of osteoblasts. And you can see that cells, even in the presence of culture media, when they are on graphene, they start expressing this protein. What you don't see on glass, right? So certainly there's some physical effect here because we don't use any osteogenic media on this group here. And when we use osteogenic media, then the differentiation goes high very, very much. So it's very interesting to see that graphene with or without osteogenic media induces some level of osteogenic differentiation in periodontal ligament stem cells. We have been puzzled by this, and we decided to run an experiment to confirm that it was indeed a spontaneous differentiation on graphene. So for this experiment, we have culture cells on glass and cells on graphene. And every other day, we were transferring media from one group to the other. So that means cells on glass will give to graphene, and in another set, cells on graphene would give the media to glass. And what we have observed here was very interesting. When we focus on osteogenic markers here on the top, you can see the increase in these green bars here, the increase of the expression of osteogenic markers when they were treated with media from the graphene group. So this happened for every other group or every other gene we have analyzed, but most prominently on the osteocalcin. So you can see that the expression goes up. And for odontogenic genes, the expression goes down. So that means that when you put a dental pulp stem cells on graphene, this cell preferred to become an osteoblast instead of an odontoblast. And this was also confirmed at the protein level, where you can see higher expression of OCN and OPN on graphene compared to glass. And keep in mind, these were cells cultured with basal growth media only. There was no osteogenic induction. So it was very interesting to see that the cells were indeed increasing their expression for osteomarkers. We have checked this in vivo as well by using a graphene scaffold that we put in a mouse. And what we have seen after 28 days was the formation of these osteo-like structures here and positive expression of RUNX2 and OPN. These are traditional markers for osteogenic differentiation, and also human mitochondria to confirm that these were indeed our cells and not cells from the mouse. So in this work here, we have unveiled the pathway that triggers the cells when in contact with graphene to just become osteoblasts. So it's when the cells are in contact with graphene, they find these structures and then they attach to the surface and they try to move the surface. And graphene is very stiff, but yet flexible. So this stress here make the cells to activate the FAK axis, integrin FAK axis, that culminates in the production of osteopontin and osteocalcin. So in this work, we have all the blockers, all the Western blots showing that this is actually the cascade of events. But one thing is, of course, is to do this on glass. So what happens when we do on titanium? Because we know that titanium per se has some properties that allow the bone cells to terminal differentiate them and also to mineralize. So here we have used two types of graphene. So the graphene is actually the same. The technique that we use to coat the titanium was either a wet technique or a dry technique. And what we have observed in this work here is that regardless of the technique we use, either the wet or the dry, there's an increase in the expression, significant increase in the expression of all markers that we have studied compared to titanium itself. And most important is the number of calcium moles found per cell in relation to the nanograms of DNA, that you can see there's almost like 3 times more calcium deposition on graphene coated titanium compared to titanium itself. So this was very interesting for us to see that graphene could indeed improve the osteogenic potential of titanium. And moving forward, we put our implants on the occipital bone of rats, and after 8 weeks, 4 and 8 weeks, what we have observed is that for the control, we still can see here the coin through the membranous osseous membrane that was being created. But for the both graphene techniques we use, we cannot really see where the coin is. And when we do the characterization after 4 weeks, you can see a lot, 8 weeks, you can see a lot of bone formation on graphene coated samples that you don't see on the control. So when we look at the healthy implant where everything goes well, we can see that graphene coated titanium, a graphene coating on titanium, let's say, can increase the speed, can increase the amount of bone formation. So what happens when things go wrong? So what happens when, if you have the biofilm there, can graphene offer some benefit for the implants? So what we have seen in our lab in 2018 was that when we coat titanium with graphene nanocoating, and we cultured either S mutants or even the whole saliva, right, by soaking the sample in whole saliva, after 24 hours, you have very little biofilm formation on the graphene coated specimens. And this study here that we have done was showing that you don't have here any bacteria dying. And that's very important for me, because I'm not looking for a strategy that eradicates biofilm, but maybe prevents their formation or delays the maturation. So then the patient has more opportunities to clean. And because we are not killing bacteria, then there's no chances that we can be inducing the most resistant bacteria to survive. We have done later with more strains. And in this work here, we have not only tried the gram-positive, but also the gram-negative PA here, the aeruginosa, right? So that's a very hard bacteria to eradicate. And also Candida albicans, because there's some literature now suggesting that Candida albicans may be implicated also on the development of periimplantitis. And what you can see here at this middle column here is very little biofilm formation compared with the control. The initial conditions are exactly the same. And here is when we quantify the biovolume of these biofilms, you can see a significant decrease when titanium is coated with graphene. We were very glad to see the results, but we were even more happy to see what was happening with PA, because this is a gram-negative bacteria, and gram-negative bacteria are very resistant to antibiotic treatment. And 90% of them are pathogenic. So in this case, where we can decrease the chances of these bacteria to attach and proliferate on graphene, you may have even decreased the need of the patient to undergo antibiotic treatment. So what was causing them, as you could see before, we are not actually killing the bacteria. So what could be the reason for that decreasing the biofilm formation? It could be related to surface changes on surface wettability. And in fact, as you can see by here, by what we call TGD, these are like double transfer graphene, you increase the water contact angle quite substantially from 70 to 110 degrees, but you decrease the surface free energy by quite a lot, from 38 to only 13. And this makes very, very difficult for the bacteria to really harbor themselves on the material substrate. And if you don't get the first colonizers, then you don't have the maturation, the growth of the biofilm by itself. So how about fungal infections? We have seen this, like there is a decrease on the fungi there, and we were very interested because fungi, they just dominate the whole body, and it's so difficult to get rid of them. And this is what we see on titanium. If you check on the titanium side, you're gonna see the hyphae formation, right? That is the virulent form of the Candida albicans. So you have this very classical transformation from the cocci to the hyphae form. But on the graphene nanocoating, you rarely have this happening, right? So you, in some aspects, they start transitioning, but it's still like way behind the mature state. Is this a transient effect? Is this because, of course, these results here is after 24 hours of characterization. So we have actually follow up these cultures for 7 days. And what we have observed is, even if you allow these biofilms to mature up to 1 week, they rarely develop on the graphene coating substrate. So on the titanium, they just keep growing because they have successfully adhered in the initial stage to a certain point that this biofilm is so thick that it's just becoming impossible to focus on the whole surface of the substrate. Meanwhile, on the graphene coated sample, the biofilms is still trying to develop and get some traction. So you imagine now a situation where the patient has so many more opportunities to get rid of this biofilm on the initial stages. That's what we are looking for with the graphene nanocoating. So how about mixed biofilms? Well, we know that biofilms in our mouth or in our bodies, they are not really formed about like a single strain of microorganisms. We are unveiling what happens with mixed species biofilms, and for these mixed species biofilms, we actually have selected one of the worst. That is the co-infection of Candida albicans with methicillin-resistant Staphylococcus aureus. And when these microorganisms are able to form a biofilm together, you can have a lot of we are not even talking about loss of implants, but even about death, right? And in this work here, we use both a clinical strain and of Staphylococcus and a methicillin resistance version mixed with Candida albicans. And what we have observed is that these microorganisms form a very dense biofilm on titanium. But when this titanium is coated with graphene, again, they fail to establish a mature biofilm. And in this case, you can see the virulent form of Candida is very weak compared to the panel on the left. And this is what we see under the SCM. You have the formation of some biofilms, of course, because the synergy between these microorganisms is very high, but nothing that is comparable with the control, right? In the control, you have a lot of hyphae formation and so many bacteria aggregated, whereas on the graphene nanocoating, these things just like really start forming after 24 hours. So if you think about a situation where you can deal with a biofilm on the very initial stage because you just delay the attachment, and by delaying the attachment, you delay the biofilm formation, this is very, very beneficial. So in these 8 years or so, we have shown that our graphene nanocoating can decrease the biofilm formation from bacterial strains, from fungal strains, and also when these strains are put together. And mostly important is that we are not like eradicating biofilms using antibiotic or antifungal drugs, but mainly delaying the attachment, and by delaying the attachment, delaying the biofilm formation and maturation. So in this case, we can think that graphene is an interesting solution that can improve titanium implants, either on the healthy side, when they increase the osteogenic differentiation and bone formation, or in the unhealthy side, by giving more opportunities for the patients and for the clinicians to deal with biofilms before they start becoming very virulent, before they start really destroying the soft tissues and the hard tissues as well. So one of the things that I want to share with you is that graphene has become like a very hot topic in research. And nowadays there is this emergence of industries selling graphene around the world. And although we welcome these initiatives because it will allow for the popularization of the material, there's a lot of concern as well on the quality of the materials that are being provided. And in these two works here from some of them here from the National University of Singapore as well, there was a big noise because our researchers have characterized graphene from 30, 40 different suppliers from around the world. And they have proven that most of the graphene material being sold worldwide is not graphene, is, let's say, graphite, extremely poor quality. That, of course, doesn't apply for all the industries, but most of them were still the quality is not there. And we actually have done some studies by ourselves and characterized the cytocompatibility of these materials. And what we have found is, even though there's a lot of discussions on is graphene cytotoxic, is graphene not cytotoxic, right? There's always these discussions like, can we use graphene for biomedical applications? What we are showing this study that is now under review is that when we buy graphene from around the world, we are not testing graphene's toxicity because we are not even having graphene there. So what we see there is that there's so many impurities in these materials that when we say is graphene cytotoxic, what we should actually be saying is graphene with too many impurities. Some of these materials, we have done some extra purification on our own. And by doing that, we have reduced the cytotoxicity by quite a lot. So there's a lot of correlations we have done in this study from this graphene that 36 materials we purchased from around the world. And what we have observed here was there's a lot of correlations between physical and chemical properties, of course, with cytotoxicity, but most important are the impurities in the materials that we have bought. So as a take-home message, I would say that whenever you're engaging on graphene research for biomedical applications, and make sure that you go for good suppliers and perform good characterization of the materials. I suggest you to do a purification step on your own to make sure that whatever graphene you claim to be using is really graphene and it's clean of impurities, so your cells or even whatever application you're going to have will perform well in contact with the graphene. So I thank you very much for having me here, and I want to thank the IDR Southeast Asia for the invitation. And you may find my profile in the Faculty of Dentistry NUS if you want to enter in contact for any clarification. Unfortunately, I cannot be here live with you now, but it's my pleasure to contribute with our society and with our members. And please enjoy the conference.
Thank you very much. I think we can go straight to second speaker on this symposiums, and that is me. I like to straight away to present my presentation today on innovation in material biology interfaces in dentistry. I'd like to share my presentation with can you see all my presentation? Okay, good morning, everybody. And I'm very happy and glad to have here in the 35 annual meeting of IDR Southeast Asia Division. And very delightful to share our research here in Indonesia in Trisakti University about the Pandanus conidus lum, or we call it red fruit extract on whole cell line apoptosis induction. Our background in research of the Pandanus conidus lum since global issues, one of Asia's most common malignant cancers, there are nearly 274,300 new cases about the malignant cancers, especially in oral each year, that we have to concern in dentistry as well. And nasopharyngeal carcinoma and tongue squamous cell carcinoma are the most common malignants in the head and neck. So early diagnosis and good treatment will improve the survival rate and quality of life of the patient. That's the reason of our background, why we choose natural plants to have in our treatment to improve the survival rate and also the quality of life. And I'd like to introduce maybe some of the research also know about the pandanus conodus lum as a red fruit. As our research here that consists of phytochemistry of a alkaloid, flavonoid, also saponin, and most consist of the physical of phytochemistry is beta carotene. And the research of this phytochemistry, which is activities of the pandanus conodus, is antioxidant. They have the antioxidant activities and also help regulate the immune system in the body and help also regulate cell proliferation. Some also research we will discuss in discussion, which is many of research in cancer already been done. So in this research, our aim of the research, we like to have how about the ability of the extract of pandanus conodus lum in the extract ethyl acetate to induce apoptosis in HALL-1 cell lines from the intercellular pathway of the cleave caspase 3 activation. Actually, this is induced apoptosis, intrinsic and extrinsic pathway. And we have also known that the characteristic of the cancer cells, and we also want to know what is the target of the therapy with the pandanus conodus lum. So the important in the cancer treatment, we have to really understand about the organelle and also about the apoptosis, intrinsic and extrinsic pathway. If in intrinsic will be due to mitochondria, and due to the mitochondria, the target in cell therapy will be succeed, hopefully can be prolong the life of the patient. So in this research, we have the identification and determination of the research plant. Actually, our research plant, we bring it from Papua, and we extract it and we macerate it here actually in Bandung. We have together work together with from our ITB person at the lab over there, and the determination and identification the plants we are going to use for the HALL-1 cell line is the real for the pandanus conodus lum. And the method that we are bringing for the research, we will do first cultivate the HALL-1 cell line. And after we cultivate the HALL-1 cell line, and then we do have the treatment for the cell line to adapt in the situation, so we can have the treatment for the with the pandanus conodus. After cell treatment, we do the harvesting the cell, and then we do preparing for the protein which we are going to extract from the HALL-1 cell line. And after we do protein preparation, we do the alphaphoresis and then transfer it to the membrane. And we do immunoblotting for 6 hours, and we do the Western blot analysis. In Western blot analysis, we have the reaction of the apoptosis and from the caspase-3 and caspase-8. And the method also, we try to do see the viability of the whole cell one HALL-1 cell line. How is the viability after with the MTT assay with the pandanus conidus lung? And we get the research that after the Western blot analysis in HALL-1 cell line, they have cleave caspase-3 activation, and cleave activation, caspase activation, we can see it from the band of the beta-actin. That is, we can compare from the positive and negative control with the pandanus conidus along with different concentration in 10, 20 ppm and 40 ppm. We can see the thickness of the band, and that is the protein which is can make the picture how is the reaction after the giving of the PCL to the HALL-1 cell line. And the result of the HALL-1 protein counting and caspase-3 cell line cleave, we can see it on the bar diagram and beside of the on this right side that we can see it cleaved caspase and the doxorubicin from the treatment chemotherapy, usual chemotherapy that they are using. We can see it, the protein density a little bit more thicker, but on the pandanus conidus, we can see it, half of the doxorubicin, around 325, and we can see it from doxo, the positive control, they have doubles, but we can see that the beta-actin, beta-actin almost the same, the cleaved caspase may be better on the doxo, but on the beta-actin, the protein, it's better, almost the same with pandanus conidus alum compared to doxorubicin. And we can see it also, although thinner concentration or lowering concentration, also the pandanus conidus also have, beta-actin protein shown. But in the cleaved caspase, there's no action in the cleaved. So apoptosis is not running on the low concentration, but higher concentration for the 40 ppm, much higher, almost the same with the positive control. As we compare with the viability of the HALL-1 cell line, which is in the doxorubicin, we can see there's significant differences. And then control and the 40 microgram per milliliter, also they have differences. And we can see it from the DMSO 40 milligram also. So there's no differences with the others, HON and other concentration for hours. This is a viability comparing in hours. So actually there's no significant difference, but only difference on the 6 hours, 12 hours, and the doxo and negative control and DMSO. And as we see the result of the research, which is apoptosis intrinsic and intrinsic play the big role, and the nasopharynx and the HALL-1 cell line, we can see the caspase-3 activate by the intrinsic pathway, which is BIT and BAX also reactive, to the mitochondria, and then there's cleavage of the bit, and then activated the cytochrome C, and then HALL-1, and then caspase 9. And after caspase 9 activated, so from the extrinsic pathway, the caspase 8, which is also received from that receptor of the outside, and the TNF-RI, FRS, and then adapter also recept all the from the extrinsic pathway, so can activate the caspase 8, and then caspase 3 also produce, and the apoptosis happens. That is the protein which is also shown in the research result on the intrinsic and the extrinsic pathway. And the mitochondria, protein of cytochrome C from the mitochondria, can activate the BCL2 family members, and then HALL-1 activate to activate caspase 9. As the caspase 9 activated, also from the extrinsic, the TNF receptor family, which is on the extrinsic shown before, on the picture before, and the caspase 8 also activate, and from extrinsic and intrinsic, the apoptosis can be shown and can be happen by cell shrinking, and then membrane bubbling, and then DNA damage, and that's the apoptosis happen. And this oral cancer is one of the 10 that cause of death, especially on nasopharynx and squamous cell carcinoma commonly found in tongue. So in dentistry, I think oral health also can play the important. And then for the most commonly found in the head neck, as I mentioned before, found 90% of oral cancer is from the nasopharynx. So we have to seek for the better and lifelong of the patient safe, and then also more curable. And actually for the cancer, that to make cell happy, it's better to cure cell cancer, and then also react to the other cell. So I think it's better research and treatment necessary for low side effect to the healthy cells. That's the main issues that why we most research in the natural plants or natural sciences. I think this will be one of the two answers how we can go through the treatment and save, also not give too much damage in other healthy cell surrounding the cancer. The natural materials that are effective and safe to be continued with adequate research. That's why we always do many research, and actually for Pandanus conioideus lung, some research already research like for the cervix cancer or other cancers, and that proven that the material are safe and have biological effects. And the main thing for the natural plants, they have what we call it with the side effect elemental substance. So the one substance to other substance in the natural plants can act to save, to defect, make a large defect in other cell on other cell health. As I mentioned in the previous slide, that Pandanus conioideus, they have antioxidant, and they have also help in the proliferation of the cells. So if the cells, the healthy cells have the apoptosis, and also including with the healthy cell, and the proliferation helping that Pandanus conioideus can help to grow with large effect. So pandanus-conidine with physical properties such as antioxidant, high provitamin A activities has been proven. As an anti-proliferation of malignant cells, their activity much higher compared to doxorubicin as drug of choice for cancer treatment in chemotherapy. So I think for this discussion, very interesting that we can more elaborate the research. The research of HALL-1 cell line viability decrease area in line with previous research. That is pandanus-conidine except fraction have cytotoxic effect and anti-proliferation activity against HeLa cervical cancer cell, lung cancer A549 cell line, and K562. And this is how the research doing apoptosis induction determination, that is with the Western blood to detecting and comparing the protein targets, and beta acting as a housekeeping protein, act as positive control or internal loading control, confirmed with equal loading protein charge. So from the protein target analysis, so we can see the cliff of caspase-3 with detected in 7 to 19 kilodalton molecules. The thicker the protein bands, the more protein target expression exposed. So in the Western blot line HALL-1 analysis, protein target cliff caspase protein target detected on 40 microgram milliliter with the ethyl acetate extract of pandanus-conidine and positive control. At contrary to negative control and 10 milligram and also 2 microgram per milliliter protein target caspase and cleaved caspase-3 undetected, as I mentioned on the previous slides. And the protein beta-actin thickness and most the same, the equal density to protein. And this is how the apoptosis shown from the mechanism, biochemistry, and morphology, which is there's a cell shrinking, there's a bubbling of the cytoplasm, and then loss of its symmetry, and loss of adhesiveness to cell membrane, fragmentation of the nucleus, chromatin condensation, and also the chromosome fragmentation. So if the fragmented happens, so apoptosis is happening, and happen by the caspase enzyme activity. And homeostasis of protein pro-apoptosis and anti-apoptosis in equal product will help caspase activity. Two different pathways can work together, intrinsic and extrinsic, in caspase activation to apoptosis. And this is the intrinsic pathway. Mitochondria induct the apoptosis reaction through cytochrome C and protein family BCL2, and then protein family BCL2 through apoptosis with BAX and BAC dimerization and entering to mitochondria membrane, and cytochrome C others and other protein mitochondria continue to cytosol. And the cytochrome C with other cofactor activated with terminal caspase-3. And the extrinsic pathway, cell expression at the molecular surface, receptor activate the receptor of TNF, and then the TNF receptor family activated also caspase-8 and go through with the caspase-3. And granzyme B cytolytic T cell product activated and caspase-3 activated. And that's the other pandanus-conidine research result on other researcher. They do for the cervix cancer to the cascade cell line, which is also from intrinsic and extrinsic pathway, which is involved caspase-8, bit cytochrome C, and caspase-3, almost the same with the HALL-1 cell line. And main pathway of the apoptosis with caspase-dependent activities and with caspase-3 activations. And the conclusion on this research is pandanus-conidine apoptosis induction depends on the concentration and time through cleaved caspase-3 HALL-1 cell line activation. And I would like to thanks acknowledge to our colleague from the Faculty of Dentistry, Trisakti University, Dr. Dicha Yulia Dewi and Dr. Vita Anggreni. And hopefully that this my presentation can bring more insight to researcher and dentists all over region and hopefully all over the world. Thank you very much and have a great day and blessed day. Stay safe. Okay, that's my presentation. We can go through with the third presenter, Professor, I think Professor Chun Chang or Po Chun Chang already with us, but I heard that Professor Chun Chang also give the presentation and video, so I can give to present the video for Professor Chun Chang. Professor Chun Chang is, I think, already introduced by Professor May Wong. Yeah. Professor Chun Chang is currently the Director of Graduate Institute of Clinical Dentistry, School of Dentistry, National Taiwan University, Taiwan, Taipei. He obtained his PhD at University of Michigan, Ann Arbor, MI, USA, and specialized in periodontology at National Taiwan University, Taipei, Taiwan. Professor Chang is a well-known researcher in the field of tissue engineering, regenerative medicine, periodontology, and implant dentistry with publication and in reputable journals. He is the editor-in-chief of the Journal of Taiwan Academy of Periodontology and the Journal of Periodontics and Implant Dentistry. Moreover, he is the chair of the publication committee, Taiwan Academy of Periodontology. He also contributes to the ITI section in Taiwan as a study club coordinator. The presentation may be, sorry.
Thanks, Chairmen, professors, doctors, ladies, and gentlemen. It's a pleasure and a great honor for me to share several biomimetic research in the IADR Southeast Asia meeting. I'm Dr. Po Chun Chang, a clinician scientist at the National Taiwan University. Biomimetics, as shown here, is a kind of the science that mimicking the structure and the function of native biological system. To be more specific, biomimetics is to design an environment not just looking like nature, but should working like nature. For the village regeneration, the biomimic environment can be divided into a macro level and a micro level. At the macro level, a stabilized and undisturbed macro environment is a prerequisite, and it can be achieved by placing a barrier membrane, bone substitute, or a solid scaffold. Infection should be also minimized to prevent further destruction and ensure the uneven healing process. At the micro level, an interface with good cell affinity favorable for cell growth is a must. In addition, the environment should provide signal or cues either osteoconductive or osteoinductive to promote bone formation. In the following slide, I would like to share 4 aspects of the biomimetic research from my research team, including to design a biphasic scaffold mimicking the periodontium, developing a functional liquid membrane by justifying the conventional barrier membrane, making a 3D printed hydroxyapatite scaffold, and the introduction of an artificial extracellular matrix. Our first biomimetic attempt was to develop a biphasic scaffold mimicking the structure and the environment of the periodontium, including the periodontal ligament, the ligament or the soft tissue phase, and also an available bone, the bone or the hard tissue phase. as hydroxy apatite plays an important role in modulating mineralization and is also a main inorganic composition of the alveolar bone. We use hydroxy apatite as the main component and use the gelatin as the binder to mold the bone face. And to make the scaffold, Chirogel technique here was used. Procedure is really simple and very straightforward. Like what we are showing here. The crosslinker was added to the uncrosslink solution and the crosslinking took place at the sub-zero temperature. In this condition, several ice crystals can be formed within the scaffold area. And then we lipolyze to remove the ice crystal. Finally, we can get a very highly porous bone compartment here. The porous structure allows cell ingrowth and vascularization, and this scaffold is also very flexible to adapt the physical and the physiological environment. To further formulate a biphasic scaffold, we then partially immerse the bone face scaffold in the cross-link gelatin and then cross-linking again at the sub-zero temperature. Ice crystal form, we remove the ice crystal, trim the scaffold, and finally we can get an interconnective biphasic scaffold with the bone face and the ligament face. Here is an example of the biphasic scaffold, and as we can appreciate here, the scaffold is very highly porous and is very flexible with a very distinct bone and the ligament interface here. The second attempt was to make a biomimic barrier membrane, namely the functionally graded membrane, FGM. We made two justifications on the surfaces of the conventional corrugated membrane. So firstly, we spread out the PDLA nanofiber on the surface of the membrane to fabricate a nanofibrous surface in order to mimic the cell extracellular matrix interface. And secondly, we loaded amoxicillin, which is a kind of an antibiotic, in the nanofibers to control the postoperative infection. Here is the resonant SCN image of the PDLA amoxicillin nanofibers network. And the mean diameter of the nanofiber was around 800 nanometers. In vitro release profile show that the amoxicillin sustained release from the nanofibers over a period of 1 month. We then check the behavior of the periodontal ligament cell, the IPDL, and the gingival epithelial cell, CA922, on the nanofibers. Both cells were well attached on the nanofibers, and the nanofiber and the amoxicillin loaded nanofibers selectively promoted the cell viability and the metabolism of the IPDL. Furthermore, as shown in the microfigure F and G, the amoxicillin loaded nanofibers significantly inhibit the commensal oral bacteria, including S. sanguinis and the P. gingivalis. The membranes were then delivered to the red periodontal defect for up to 1 week, although we did not observe obvious difference in the periodontal bone gain. Mucosal wound healing was promoted, and the level of the circular inflammation was reduced by using the PDLA amoxicillin membrane. Gene expression profile here demonstrates that in the PDLA amoxicillin group, the inflammation-associated gene, including the IL-1 beta and the AMPA, both were downregulated. And we can also appreciate that the matrix synthesis gene, the HALL-1, and the angiogenetic marker, the VEGF, both of them were upregulated in this group. To make our FGN more functional, we made the both surface of the membrane the functional layers. The concept is that on the outer surface, we encapsulate metronidazole to prevent the infection. On the inner surface, we encapsulated the PDGF in the nanofiber to promote cell proliferation and facilitate the healing and the regeneration. So here are the nanofibrous functional layer on the membranes. In vitro release profile show that both PDGF and metronidazole were sustained release, and the PDLA PDGF group significantly promote metabolism of the PDL cell at the 24 hours, the day 1 data. showing here. We deliver the membrane to the spiraglotally rat and check the pCNA, a cell proliferating marker, by immunohistochemical staining. We can appreciate here the PDLA PDGF membrane promote the cell proliferation, very prominent here. And on the alveolar ridge defect, the empty group, which is the one with the metronidazole, PD group, the one with the PDGF, and the MP group, the one with the PDGF metronidazole combination, promote the early mucosal wound healing. The micro-CT image here also show that the PD and MP group had the greater amount of the osteogenesis in the defect. However, quantification data from the micro-CT and from the histology show that only the PD group, which is the one with the membrane with the PDGF alone, had significantly greater bone formation compared to the control CT group and the membrane alone group, the CL groups. This is a further study on the dog. And in this study, we try to combine the biphasic scaffold with the FGN. Based on the previous study, the choice of the FGN was the one with the PDGF or loaded membrane. And regarding the biphasic scaffold, we not only we made the biphasic compartment, but we also added the biomolecules, including the EMD, enamel matrix protein, in the ligament phase, and the BMP2 in the hard tissue phase. The procedure is looking like that. So we create the defect and then fill the defect by the biphasic scaffold, cover the scaffold with a membrane, and then do the primary suture. After 12 weeks, our data show that either the biomolecular loaded biphasic scaffold or the FGN promotes osteogenesis in the defect. And from the histology, the results show that the FGN reduced the CEJ gingival sulcus distance, which can be referred to a greater pocket reduction in humans. Histologic evidence also show that when the EMD were loaded in a biphasic scaffold, functionally oriented periodontal ligament fiber insertion with a very obvious new cemented formation on the denuded surface could be observed. In the specimen without the EMD, the ligament just run parallel to the root surface without obvious cemented formation. Here is our latest approach on the FGN. In this part of the study, we made the nano composites in which functional nanosphere were embedded in the nanofiber and then adhering on the surface of the membrane. And both the nanospheres and the nanofiber can encapsulate with the biomolecules. Idea is really similar as what we described in the previous slides. We still made the outer surface and the antimicrobial layer, and on the outer surface, we encapsulate another antibiotics, which is doxycycline. Unlike other antibiotics, doxycycline not only inhibits bacteria, but also an inflammation modulator as well. So using doxycycline, we may control both infection and inflammation at the early healing stage. The doxycycline can be released when the nanofiber was degraded. On the inner regenerative layer, we made the EMD-loaded chitosan nanosphere and then embedded in the doxycycline-loaded PDLA nanofibers. So the concept is that the doxycycline are still first released from the nanofiber, and then the nanofiber are degraded to expose the chitosan nanospheres. And then the EMD can be released from the chitosan nanospheres. Here are the validation data. On the outer surface, our data show that the doxycycline-loaded nanofiber significantly inhibit the P. gingivalis and inhibit the S. sanguinis. On the inner surface, basically the nanospheres were evenly distributed within the nanofibers. And this configuration also slowed the release of the encapsulated molecules from the nanosphere, and the EMD encapsulated nanosphere in the nanocomposite promotes the alkaline phosphatase activity for the in vitro mineralization. Preclinical data also demonstrate that the AMPA, the tissue degradation marker, was downregulated when the doxycycline was added. As we can appreciate, these two groups was downregulated. And the ROX2, the osteogenesis marker, was upregulated when the EMD was loaded, as we can appreciate in these two groups. The micro-CT data also show that the specimen of the EMD loaded and the EMD doxycycline combined nanocomposite significantly promoted the bone formation, as shown in the BVTV. And this configuration can also achieve the greater trabecular thickness, which means that the newly formed bone appears to be more corticalized compared to the control group at 4 weeks. All data here support that the justifying the membrane with the nanofibrous layers loaded with the antibiotics and the biomolecules is a valuable biomimetic option for the alveolar ridge regeneration. The next aspect is the 3D printed scaffold. There are several major advantages of making a 3D printed scaffold, including we may better control the scaffold architecture and the property. And we may customize the scaffold based on the medical imaging of the patient and design the scaffold using a computer-aided software. This digitalized workflow makes things easier, faster, and usually cheaper than the conventional methods. There are several specific requirements for fabricating a 3D printed scaffold, including the scaffold, the material should be biocompatible, and the scaffold should provide a biomimetic interface. The scaffold should be chemically stable and should be capable of delivering the cells or the biomolecules. My team chose hydroxyapatite as the base material to print because hydroxyapatite was chemically stable and could provide a biomimetic interface favorable for osteogenesis, and it is also the main composition of the human bone. In order to preserve biocompatibility and to prevent potential thermal or chemical damage, an extrusion-based printer was used, as shown here. And the bio-ink we use containing 90% hydroxyapatite with 10% biodegradable polymer, PLGA, and the PCl. The function of this polymer serves as the binder to mold the scaffold. We use a CAD software to design a porous structure like this, converting to the G-code and print out. And here is an example of the resident scaffold. As shown here, we can appreciate that the size, geometry, and the porous structure was generally stable and very homogeneous. We validate the efficacy of the 3D printed scaffold using two animal models. The first model was the jawbone intrabony defect model, as shown here. So we surgically created a through-and-through intrabony defect on the red jawbone and filled this defect using the 3D printed hydroxyapatite scaffold. The result here are the micro-CT data showing that compared with the on-field control, we can appreciate that the scaffold or treating the defect were mostly occupied with the mineralized tissue. Histology also showed that there are still a certain amount of residual scaffold occupied within the defect area. But after we subtracting the residual scaffold, the remaining the newly formed bone was still significantly greater in the treated defect compared with the control group here. And we can also appreciate very good the tissue and scaffold and the bone scaffold interfaces. We further check the efficiency on the supracrystal regeneration model. And in this model, we make the scaffold and place the scaffold supracrystally on the lateral surface of the jawbone. And we fix the scaffold using a mini titanium implant as shown here. In this part of the study, we also compare not only the on-field control, but we also compare the efficiency with the one filled with the DBBM, which is the deproteinized bovine bone matrix. The result here are showing that basically the DBBM group and the scaffold group have the comparable quantitative outcome in either the mineralized tissue formation and the newly formed bone formation from the micro-CT image. However, in the DBBM group, we can appreciate that several specimens reveal the spread out of the particle, and the vertical bone formation was relatively limited compared with the control group. We can also appreciate that in most of the specimens, regardless of the treatment group, there was no newborn formation on the top level, and most of the specimen only demonstrate the newborn formation at the crystal level, even after 8 weeks of the scaffold placement. So in summary, this study supported the osteoconductivity of our 3D printed scaffold. However, to achieve the regeneration in a condition with limited regeneration potential, such as the supracrystal area, we may still have to consider the exogenous source of the signals or the cells. The 3D printed scaffold, as shown in the previous slide, provide a stable microenvironment and a biomimetic interface. To modulate the cell behavior toward the osteogenesis, we also care about the cues from the microenvironment. To be more specific, cells are surrounded with the extracellular matrix. Would the property of the extracellular matrix affect the differential potential of these cells? So to answer this question, a group in Harvard University work on how the stiffness of the extracellular matrix modulate the cell behavior, and they use alginate as a base material and manipulate the stiffness by controlling the extent of the cross-linking. The result basically are showing that the stiffness did direct the cell differentiation. A harder matrix, especially when the stiffness was equivalent to the osteoid, which is about 20 to 40 kilopascal, may favor osteogenesis. When the matrix is softer, especially when the stiffness was equivalent to the mucosa, may favor adipogenesis. The result inspire us and come out a hypothesis. In a stabilized microenvironment provided by the 3D printed hydrostatic scaffold, adding an osteo-equivalent artificial matrix to direct the cell toward the osteogenesis could be a beneficial approach for the alveolar regeneration. To test the hypothesis, we use alginate matrix to formulate an extracellular matrix, and the matrix is also functionalized with RGD sequence because without the RGD sequence, the cell cannot recognize and cannot attach on the alginate matrix. So the alginate matrix was injected to the 3D printed scaffold as shown here. The matrix had a different stiffness, and we oxidized the matrix to make them degradable, such that the cell and the vessel could invade to generate the new tissues in vivo. As we can appreciate here, the swelling ratio demonstrates that the oxidized alginate matrix, which is ORO group, degrade much faster compared to the non-oxidized ones, which are the NRO and the NRL groups here. We then implant the scaffold with the matrix to the jawbone intrabony defect. So basically, the micro-CT image is showing that in the specimen with the scaffold, all of them, the image was comparable. Most of them demonstrate completely filled with the mineralized tissue. But after we subtract the residual scaffold in the tissue area, we can appreciate that the SCORO group, which is the one with the degradable oxidized alginate matrix, have a significantly greater amount of the new bone formation and greater bone bridging compared to the control or the scaffold alone group, the SD group. Histologic imaging also show that in the SCORO group, there are more new bone formation and more new bone infiltrating to the defect area compared to all the other groups. As the scaffold with functionalized degradable stiff matrix show a very good intrabony outcome, we are now running a project investigating the efficiency on the supracrystal area, and consider the inferior regeneration capability of this area. In this part of the study, we mix the bone marrow stem cell in the matrix, infuse the cell-loaded matrix to the scaffold, and then fix the scaffold complex on the jawbone, which was the same procedure as I described in the previous slide regarding the supracrystal ridge regeneration model. We are still in the middle of this project, and the current data show that the SCORO group, which is the one with the scaffold with the biodegradable and the stiffer cell-loaded matrix, had significantly greater osteogenesis compared to the control and the scaffold group, the SD group, at both 2 weeks and 8 weeks. The histology showed that at 2 weeks, the newly formed bone was limited to the border in the SCORO group, and there are still a very significant amount of the residual alginate matrix within the scaffold area. However, at 8 weeks, we can appreciate that most of the matrix was already disappeared. And there are significantly more bone formation and infiltrating into the scaffold area. And there are also the initial bone marrow space formation in the SCORO group. So there are 2 hints here. So firstly, 3D printed hydroxide scaffold with artificial matrix can be a workable solution for facilitating the alveolar ridge regeneration. However, more research regarding how to harmonize the degradation and the stiffness of the matrix, as well as investigating the interaction between the degrading matrix and the cell, should be performed in the future. Secondly, in the supracrystal region, supplement of the stem cell or exogenous molecular signals may be still required. By the end of my presentation, I want to acknowledge the support from my collaborator, staff, and students. I also want to thank the funding agency listed here. If any of you have any question or comment, please feel free to ask or to provide. Thank you for your attention.
Okay, thank you, Prof. Chun Chang. I think we come to the panel discussion session. I'd like to open the question and answer for all the participants. If you want to ask or discuss about what we share this morning, you can ask straight, or you can write down to the chat box. Professor Po Chun Chang, can you open the video?
Hi.
Hi.
Hi. Because I don't have the camera in this room, so I'm sure that I cannot open, but I'm here. So any question, please feel free to ask.
Yeah, okay then. I would like to Prof. Chun Chang, as you mentioned about your research that is still running, right?
Yes.
Can you give a description for the end of the research of your study here? Maybe can you describe a little bit about your study for end result, like upstream, downstream of the research result will be for the dentistry, as you mentioned about the scaffold of the development of biomimetic interfaces for alveolar regeneration, for the maybe for the implant treatment in the future, right?
Yes. Hello.
Yeah.
Actually, because as I mentioned that I'm a clinician scientist and my specialty is in periodontology and implant dentistry. So what we want to develop is we want to have something that can be used to facilitate the periodontal and the peri-implant tissue regeneration, especially like when we handle most of the time I'm handling with the patient with the deficient alveolar ridge. So I want to do the augmentation to have a sufficient amount of the bone, and then we can proceed to do the further processes and the implants. So the goal of those study was to is to develop the material for this purpose. And yeah, because although we have many materials can use here, but can use today, but most of them are still, I mean, sometimes in the area without a very good regeneration capability, it is still very challenging. So we want to see if the biomimic interface can give us a more favorable outcome, more favorable and a more predictable outcome. And as our ongoing study demonstrate that, yes, we may use the scaffold, and we may use a scaffold to provide us a very stable macro environment. We can use the algebraic matrix to provide a very biomimic microenvironment, and we also implant the stem cell. We feel this may be a solution can be more efficiently to promote the efficiency of the regeneration procedure. So, but we are still running the experiment, and I want to say that although we have the goal to make the new material, but we are still running and still try to find out the better formulation or the better ingredient, because we feel that there are multiple formation, but still are not sufficient. And we want to see if we can get more cell, or if we can further manipulate to find the most appropriate stiffness, and then to get a more favorable outcome. Thank you.
Thank you, Prof. Jun. This is very interesting. Since also Professor Rosa mentioned about the nanocoating, graphene nanocoating to improve osteogenesis and decrease biofilm on the titanium. This is also mentioned for the implant treatment, right? So I think this is quite very interesting for dentistry to the future. Yeah, I think it's better for further research in the future. I really hope that this kind of research, as my research and my colleague research as well, we do hope that this is not only like you know, a tower of most of research, but how it affect to the people, to all the people in around us. I think that's the main issues as I research and my colleague research, and many of many of Indonesian researchers doing for natural plants and natural materials to do in dentistry. They do also mention about the scaffolding with the natural materials, and they do hope that we can also in the future do together, since we are in the same region and we have the same climate, and we do this more research in the future for bringing to the better for our people surrounding us. As we are now, although having, you know, facing still in the pandemic, but hopefully that in the near future we can cope with the good health and new norm, and how we do it in the future. What do you think, Prof. Jun, about the research on the future of our research?
Hi. So do you mean that the research field, or what do we think about how our research can like inspire students?
And I think in both you can explain whether in our area or do's to what in the future be ahead, because sometimes the it's very fast in moving, in health especially. So I think from the research we can cut down from here to let's see, you are aimed to the implant, but maybe later on can also act for the for the good health for the people around us.
Yes, I agree. Actually, I think because I mean, I think although we are the dentists and we work for the field of dental research, but I think our research is not limited to solve the dental specialty problem. I mean, like now I'm working with some like the engineers and some medical doctors. So I think in the future, I mean, I think for the dental research, I think what I did is just like a beginning. But I think that we after we have this data, and then we both based on this data, we can provide some solution for many medical issue, and make also provide some some data for our engineer to see how to improve their fabrication process, or even they can make the new instrument using the new technology to not only to assist them to develop the new tech, but also the new tech can also help us to develop maybe the new treatment strategies. I believe, and I think this is also a very good for I mean, because I mean, like we just open a door for the future advancement of the clinical, of the industrial, and for the future research. I think this is also a very good for our new our student and the young scientists to further work on those researches. Thank you.
Yeah, totally. I do agree, Prof. For doing that for next research. So I think we just actually in our presentation, the three presentation and this symposium hopefully can bring more research in our field in dentistry, but also in young research, and we are senior can go along together with them, and what we can have it after this meeting, something we can go with to our own country, and maybe we can think for further research in the future. Because if I talk about in our country, we are very eager to find something new. I said that they don't have to be something new, but something more important and more easy and eligible to do it in our country, and best to our people, and not only Indonesia but surrounding Southeast Asia and Asia Pacific and all over the world, hopefully. Then we are very rich. We are very rich in our biodiversity. So I think we can elaborate and we can go together with many research in the future. And hopefully, what the technique that you mentioned from Prof. Chun and Prof. Rosa, I think this is very important for us to do collaborate in a method, or collaborate in ideas and many things in the future. Do you think like that, Prof. Rosa? I don't think he's joined with us. I think only two of us, Prof. Chun. Prof. May, I think we can conclude this symposium with a good result discussion of two of us. Hopefully all the participants can also get the benefit from this symposium.
Thank you so much, Professor Melanie, Professor Zhang, and Professor Rosa for the very excellent talks. That will really help especially our scientists to think about like what work has been done and what further work that they can thinking of, like continue the good work of all the researches that you all have been doing. So thank you so much. Let's conclude this symposium, and our next session will be our poster sessions at 12 o'clock in the poster hall 123. So see you back. Bye-bye.
See you then. Bye-bye.
Bye-bye.
Bye. Thank you.
Hello.
Hello, how are you doing, Warren? Hi, Murray. How are you doing?
You. How are
Good.
you?
Very good. Been a busy few days.
Yeah.
Do you want to try and see if you can share your presentation?
Yes, of
Warren?
course.
Yeah,
Yes.
and then we'll just sort of see.
All right. So I share it now.
Yeah, and just see does it work. I think so far things have been working out.
Mhm. Can you see my slides? And
No.
no. No.
Maybe the tech team have to do that.
Mhm.
How
I
about you,
think
Murray?
because they're sharing their PowerPoint at the moment, so
Oh,
we
so they have
can't
to stop.
concentrate on that. Yeah.
You're a dab hand at all this with all the lockdown you've had in New Zealand.
Oh gosh, yes.
You're just out of the blocks, as they say.
Yeah, yeah.
How has the
Hello, everyone.
I may.
I'm May here.
Okay.
However, I got a problem with my Zoom, so I need to log out and come back again. So
Okay.
I
sorry
was
for
wondering how you were talking without moving your lips.
yeah. That's true. See you later.
See
Yeah.
you. Maybe I try sharing my slide again. See if you can see it. Can you see it?
No, it may be that the tech team are just sharing and they have to give us the ability to do that.
Okay. All right.
What I'm doing is I'm doing a direct message to them to ask them to stop sharing their screen.
How are you doing in lockdown, Warnock? Are you locked down the school? No. You're on mute.
Sorry. No, we
Good.
don't lock down this year.
Ah, great, yeah.
Those in Bangkok, like they have to close their schools,
Ah.
but not in our region.
Yeah, good.
Wow. Yes, we had the Auckland region in a much longer lockdown than the rest of us, because a lot of them refused to behave, you know.
Hello. Here I am. Okay now.
Yes.
Yeah, thank you. Thank you for helping. Yeah.
Thank you for inviting.
I really miss meeting people face to face, in person.
Yeah.
Even though we are so used to this online meeting now, but still, I would like to see people.
Yeah.
I would much rather be in Hong Kong right now than sitting in my home office.
So what happened there?
This is where I play. So this is my piano behind me there.
I don't
Oh,
have
nice.
time to play the keyboard yet. So
And
what do you
over
play?
there-
What songs do you play?
Oh, well, over there on that shelf, that bookshelf, that's all sheet music on there. And I've got a collection that I've been amassing for 50 years.
Classical music?
So I play all sorts of things. But I'm self-taught, so you know it's not the flash classical stuff.
Okay.
It's fantastic.
I can do your Springsteen or Fleetwood Mac or Elton John or
Oh, so nice.
you name it. The
That's
great
something I
American
want to
song.
do as well.
Yeah.
So how often do you go back to office, Marie?
Go back to where?
I mean, university office. Do you have an office in a
Every day. Yeah.
Still,
yeah, no,
every day.
We're no longer in lockdown. Yeah.
Okay.
We had a big one last year, and then this year we had one for about 3 weeks.
Okay.
Down here anyway. Up in Auckland, they had one for much longer.
So how about you, Waranu? Everything
No
came
lockdown.
good?
No lockdown at all. We are fine this year.
So you can freely travel different places.
Not really. Not like in Khon Kaen, the situation is good. So, but then in Bangkok and some area in Thailand, it's not very well. So we need some precaution. But yeah, we can travel.
Okay, that's good. But you know, Hong Kong is so small.
Yeah.
When we say travel, there's not many places that we can go. But still, a lot of people trying to go hiking. And we have really many trails that you can try.
Oh.
May, we wanted to test out whether they're okay at sharing their screen.
Yeah. Now, you can try now. The tech team has, yes.
So Warna,
yes, you can check your.
You can try now to see if you can share.
All right. Can you see mine?
Yeah, perfect.
Oh, beautiful, beautiful.
Okay.
Nice.
Oh, but I cannot move my slide, so it's kind.
Really? Try.
Oh, let me let me check. Right, can you see my slide now?
Yes.
Yep.
And then.
Okay. All right.
Yeah, it's good.
Yep.
Super.
Good. Okay, great. Thank you.
Excellent. How about Murray?
Let's try this.
Murray.
That one. Can you see that?
Yes.
Yep. And does it move? Yeah.
Yeah. Yes.
Super.
Cool. Okay, so I'll stop sharing that.
I just try now and see. I think you see, and it moves with that. Yeah. You can see, yeah. Oddly enough, you don't see it yourself, which is always kind of like, so you understand why people always say, am I sharing this? I think May is going to do the introduction. We got a few more minutes anyway.
I will introduce all three of you and then pass the time to comment.
Super. Well, yeah, and Murray is the first one then. Congratulations to Jonathan on his professorship.
Yes, he's very pleased, and we're very pleased.
Sure, it's super. I'm really delighted for him. It's really good, and he'll be in well position for you when you're exit or partial exit or whatever you're doing.
Yes, yes, and you know, he's having a good few weeks because he turns 41 on Sunday. Sorry, Monday. So,
amazing, amazing.
It's a bit like your kids. I think, gosh, how did he get that old so quickly? You know.
Yeah, it's phenomenal. It's phenomenal.
Great achievement, actually, and recognition as well.
Yeah, yeah.
He's done some lovely work.
Yeah. He's you know, still got a lot of potential, so we're very pleased. I have told the university that I am retiring in Easter 2023. Because psychologically I need to have a finish line, you know. So,
but did you say you were retired 2 years back?
Well, I'll still dabble, you know. It'll probably be a John Spencer retirement, you know. Never mind.
So tech team, actually, we hear you talking at the background. So
Dipali Ajmera.
Might pay to send them a note on the chat. Tech team, we hear you. Yes, like
Tech team, we don't have control to stop them, to mute
The
them.
thing is, like, they should make us, or at least one of us, to be a
Yeah.
co-host. So if anything happens, we can control. Not like another poster session, we just got a problem and no speaker, not even the chair was a co-host. So
Yeah, I saw that the last. Yeah.
and she was so panicked. But anyway, Melody handled it very well already. So I have to say.
Prof. May,
Yes.
should we go live after 2 minutes?
Yes, sure. So Coleman, your PowerPoint was okay, right? You share it okay.
Yes, we did. We all shared.
Yeah. So we can go live in 2 minutes. Just let us know when time is time for
Okay.
to go live.
I thought Varuna had suddenly gone down. She's back up.
So one last session, and then we all bring to an end, and then we will have the awards ceremony at 4:30. So far, everything was okay.
good.
We're about time to go live. Head team, is it time to go live now?
Ma'am, you're live now?
Yes, thank you.
Professor May, you can start.
Okay, thank you. Yes. Welcome back. Welcome to our third symposium. And the title of the symposium is Oral Health and Successful Aging, Implications for Research and Practice. So today we are very honored to have three excellent speakers here, and I would like to introduce them to you. The first speaker, Professor Marie Thompson. Professor Thompson is a dental researcher and specialist in dental public health based at the University of Otago in Dunedin, New Zealand. So his work is in the renowned Dunedin study has enhanced understanding of oral health through life. He is an expert on dry mouth and has developed widely used measures for use in clinical practice and research. He has also made important contributions to understanding of oral health of the aging population and of the effectiveness of dental care in improving the lives of children and their families. To date, he has published over 400 research papers, 5 book chapters, and his H-index is 56. He is the editor-in-chief of Gerontology. He was the editor for Community Dentistry and Oral Epidemiology from 2015 to 2021. He also was, he is the associate editor for the European Journal of Oral Sciences. And the next speaker is Professor Coleman McGrath. Professor Coleman is a clinical professor in dental public health at the Faculty of Dentistry at the University of Hong Kong. Coleman is a past president of the Behavioral Epidemiology and Health Science Research Group of the IEDR. Coleman recently served on the steering committee for the 2020 Behavioral and Social Oral Health Science Summit and was the lead of the Summit Reactor panel in Southeast Asia. So Professor Coleman's research interest is on the social impact of oral health, social inequalities, and the oral health of older people. He has published over 300 papers, and he has a particular interest in improving the oral health of people with disabilities and serves on the scientific committee of the International Association of Disability and Oral Health. Last but not least, our last speaker is Dr. Waranu Patipat. So Waranu is the Dean and Associate Professor of the Faculty of Dentistry, Khon Kaen University, Thailand. He is a diplomat of the Thai Board of Dental Public Health. Professor Patipat is active in research, particularly relating to the oral health systemic disease link and epidemiology of oral diseases. So he has over 90 publications and has served in numerous editorial boards, including like JDR Clinical and Translational Research, and Oral Diseases. Professor Patipat is the past president of the Asian Academy of Prevention Dentistry and the Thai Society of Public Health Dentistry. He is currently honorary secretary of Southeast Asia, the Association of Dental Education, member of the board of directors of the Biruran Center of Global Oral Health at the University of Washington, and distinguished adjunct faculty of Savina Institute of Medical and Technical Science, India. So today, our chairman for the symposium is Professor Coleman McGrath. I would like to give the time to Professor McGrath to chair the session. Thank you. Professor McGrath, please.
Thank you very much, May, and welcome to you all to our symposium on oral health and successful aging. We'll have about 25 minutes for speakers, and at the end, we'll have the questions, about 15 minutes for questions. So without further ado, it gives me great pleasure to introduce again Professor Thompson, who is a colleague that we call upon many, many times. He is not just an expert in dry mouth, he's an expert in aging, which is just as well as he's about to be categorized as an older person. So without further ado, Murray is going to present to us some of the findings and understandings of oral health and aging, and particularly with respect to New Zealand and the wonderful life course studies there. Murray, thank you.
Thank you very much, Coleman. I'm going to share my screen now, and we'll see whether it works. So if someone can confirm that you can see my screen.
Yes.
Is that fine?
Yes, everything good?
Right. Okay. Well, kia ora, everybody. As Coleman said, I'm rapidly aging, but you know, you're only as old as you feel. So I feel like a 21-year-old tonight. So let's go for it. So it's a bit of a strange presentation I've got for you this evening, or this afternoon, your time. I'm going to be talking about presenting some of our findings about aging earlier in the life course. And then I'm going to segue to some findings from what is only the world's second national oral health survey of people in nursing homes. And then I want to come back to some conceptual issues and think about the issues that have been raised in looking at the research that we've got, that we will have looked at. So let's go on. So thinking about the pace of aging now. You know, if you think about aging, we're all getting older at the same rate. You know, every year I'm a year older, and so are you. But where we differ, where we vary, is in the pace of our biological aging. You know, that biological aging is simultaneous decline in a number of organ systems, and it's a gradual thing, but you know, it is a progressive thing. It's cumulative. You can't turn it back, however much some of us try or would like to. And then, you know, we've got this notion that we have our individual genetic differences. We also have differences in the way in which our physiology functions, if you like. And of course, we're all having different life experiences and different exposures. So all of those beneficial and adverse exposures and our physiological and pathophysiological reaction to those, that all accumulates and ages us. And so for most of the body, we have this subclinical organ system decline that actually begins far earlier in life than you know, we all think about old people as being old and aging, but we've all been aging since probably our late 20s. Oral conditions are the exception to this. You know, if we think about something like periodontal attachment loss or dental caries, we know that this accumulates clinically at a relatively constant rate, particularly dental caries. And we know from our longitudinal study that, you know, it's about one surface a year in the average person. Take a look at the picture here I've got here of these people. They are all 45 years old. And as you can see, the people on the left look considerably younger than the people on the right, who, for all intents and purposes, could be their parents. I'll come back to that picture in due course. So I'm assuming that you will have heard of the Dunedin Multidisciplinary Health and Development Study, which is this, excuse me, wonderful prospective life cohort study of a complete birth cohort born in Dunedin in 1972-73. And it's the only study really that is telling us what is happening to people, particularly to their oral health from adulthood, you know, through adulthood. You know, we've had a lot of longitudinal studies on children. We've had quite a few on older people, but this area, you know, this part of the life course in midlife is really untapped. And so the Dunedin study has made a lot of contributions in terms of our understanding of the natural history of oral diseases. And something that's really fascinating is a paper which we published this year on the pace of aging. If you have a look here, this is a very busy slide for which I apologize. There's where you can find the paper. We looked at 4 different groups of outcomes. We looked at neuroimaging, so we did MRIs, we did cognitive tests, we did sensory motor functional capacity tests, and we got people to rate these people, our study members, in terms of their aging, both their own self-rating, and we got outside observers to rate each of our study members. As the exposure, we measured a suite of 19 biomarkers at each of these ages. Excuse me, 26, 32, 38, and 45. We standardized each one on its baseline, that is age 26 distribution. And then we determined each person's personal rate of change for each of those biomarkers, and then combined that into a single pace of aging variable. And, you know, we came up with a fairly normal distribution, if you like. And we also had dental markers in that pace of aging variable. So we had average periodontal attachment loss and the number of DMFS. So if we go forward to what we found, it was essentially normally distributed with a bit of right skewness here, where these people are aging much faster. And if we look at these people and we look at their brain structure and function, for example, excuse me, I've got hay fever. You know, their brain structure was noticeably different by midlife, by age 45. There were signs of early white matter deterioration. They had a higher brain age in terms of this composite measure that was made up from these indicators. Their brain function was poorer. They had poorer cognitive function. They had more cognitive decline. They had slower gait speed, poorer balance, and poorer sensory motor function. So these people are already becoming old people much faster than the remainder of the cohort. And if you have a look at this, if we say that if you're aging at the same rate biologically as you are chronologically, your pace of aging will be 1. Our range was 0.4 to 2.4. So the person who is aging at 2.4, in terms of their pace of aging, is aging almost 2.5 times as quickly as the average person. I'm going to come back to this. Oh, hang on, let's just get back to the facial aging. These are digitally averaged composite faces made up of the 10 participants from this distribution here, with the slowest pace of aging and the fastest pace of aging. And I'm sure you'll agree with me, if you look at compare the people on the left with the people on the right, there is a considerable difference between them. I'll come back to these findings in a little while. I just want to digress for a little bit and just tell you about our work with older people in nursing homes, and then I'll come back to some conceptual issues. I told you this talk was a bit of a dog's breakfast, really, but bear with me. It'll all come together in the end. So almost 10 years ago now, we were fortunate enough to get funding from our Ministry of Health to conduct a national survey of oral health in rest homes, because we'd conducted our 3rd national oral health survey in 2009, and that specifically excluded from the sampling frame people who are living in nursing homes. And so the only other such national survey had been conducted in Belgium. So as with all of these things, there is a heck of a lot of people involved in collecting the data, and I'm very grateful to all of these people, and particularly to our Chief Dental Officer at the time, Robin Haseman Welsh, who basically secured the funding for this survey. So we assessed essentially 1,100 people who are living in nursing homes, and just under 1,100 people who were aging in place under the government's aging in place policy. So if you look at the population in New Zealand aged 65 or older, 5% are in nursing homes. Another 10% are being supported by the government with daily carers, etc., to stay in their own homes because it's cheaper and it's better for someone to be in their own home. So we were also interested in the oral health of those people. So we included both groups in our sample, and we came up with a stratified random sample from which we can generalize back to the source population of about 20,000 people. And we were able to look at things like dentition status in far more detail than previous national surveys have reported. Normally you'll see estimates of the edentulous proportion, the proportion with a functional dentition and whether they're wearing a denture, the number of teeth, etc. Natalie Hyland worked with us. She was a very bright, finally a student of ours, who we taught to write the code, and she did a great deal of work characterizing all of our dentate participants' dentitions using the Kennedy classification, so that we came up with a lot more detailed picture of the residual dentition in these people. And if you have a look at at it here. Here's the maxillary dentition, here's a mandibular dentition, the residential care population and the home-based population. And you can see that, you know, very few of them were almost completely dentate. Nobody had a complete dentition, but these people had maybe one tooth missing. A combination that's very common in, particularly in New Zealand, Australia, Scotland, England, Wales, is an edentulous maxilla against standing lower teeth. So, and you can see here that there was 1 in 5 of our sample. And so, you know, I won't go through this in detail, but you can look up the papers and have a good read of those. They are very, very fascinating. If I say so myself, of course. I want to move on from this now. We also looked at clinical oral disease status by cognitive function and dependency level, testing the hypothesis that those with poorer cognitive function would have poorer oral health. And certainly that's the way it worked out, certainly for those with severe cognitive impairment. And you can see here that their number of untreated decayed teeth was 50% higher again than people with normal cognition. Interestingly, there are no differences by dependency level, which surprised us. Similarly, for how clean the teeth were. So this is using the OHIS score. So the higher the score, the dirtier the teeth. And again, this severely cognitively impaired group had scores that were 80% higher on average than people with normal cognition. And we've done other work recently, published recent work, doing secondary analyses of the data set. So we've looked at nutrition, we've looked at drug taking, we've looked at dry mouth. And we've also, for our future research, confirmed the clinical validity of self-reported oral health among these people. So using David Locker's famous single item, you know, how do you rate the health of your mouth? And we were able to show that does have a high degree of clinical validity. In other words, those who rate their oral health as poor have much poorer oral health than those who rate their oral health as excellent. So I want to come back now and have a look at some issues. And this is really wearing my gerodontology editor hat a lot of the time too, because I see the research that's done out there on older people. It comes in and goes out to review if it's lucky, and comes back and eventually ends up getting published. But I see a lot of the weaker papers that don't make it as well. So let's have a think about some of these issues. Older people don't just appear. They don't just appear in front of you in the supermarket, pushing their trolley very slowly or whatever. You know, we have aged. We were once young. You know, excuse me. They've been shaped by what we call their journey through the life course. So there are aging effects, you know, the maturing, and then the senescence, this decline. But there are also period effects and cohort effects. For example, Coleman's generation and mine had the best music. That's a cohort effect. You know, so what we see in their oral health reflects all of those influences. And I think nobody put it better than Ron Ettinger back in 1993. Elderly individuals are a complex combination and expression of their individual genetics, their lifestyle, their socialization and environment, all of which affect their health beliefs and their health behavior. The key sentence is the second one. To understand an individual, one must evaluate the social, cultural, economic, and chronologically specific cohort experiences which have shaped his or her life. So let's think about this. You know, if we've done a survey of older people in last year, let's assume COVID didn't happen and we did a survey, we actually have people there who are very, very different and have had different experiences. You know, the 100-year-olds, the 90-year-olds lived through part of their lives through the Great Depression, World War II. Then the the 70-year-olds, if they live in a fluoridated area, they've lived all of their lives exposed to community water fluoridation, for example. If we think about a disease such as dental caries as being very chronic, cumulative, and you know, the accumulation of good and bad exposures happens very, very slowly but steadily over the years. These exposures make a difference. And the advent of fluoride toothpaste back in 1970, you know, you brush your teeth twice a day with that stuff. That's 730 mouth rinses a year that we never used to get. That's been responsible for a big fall in caries levels. And the advent of the air rotor, for example, invented here in Dunedin by our old Dean, Sir John Walsh. Composites and GIC, then implants, etc. All of these things are affecting how, you know, what we see in these people's mouths when we do this cross-sectional look in our survey. So I suppose I'm making a plug here for being careful about interpreting cross-sectional data or even longitudinal data on older people. And the classic example of this is the Periodontal Systemic Health Association. Periodontitis has been claimed to be a causal factor for all of these things. But as pointed out by Bruce Pilstrom and colleagues in a JADA editorial in 2018, there are a lot of flaws in the knowledge base. You know, most of the knowledge comes from cross-sectional studies. There's been an inadequate control of important confounders, things like smoking, poor and inconsistent characterization of exposure if we're treating periodontitis, periodontal inflammation as the exposure measure. And then there's been overinterpretation of poor data and mechanistic bias. This is the bias where if we can come up with some sort of biological explanation for it, which we've seen in spades over the last 3 decades, then that seems to give it a degree of respectability. And of course, there's also publication bias and a degree of groupthink. And the latest sort of episode in this sort of thing is the whole tooth loss and cognitive function association. So I want to spend a bit of time looking at that. Here's data from the original paper that started the whole thing off. This was a longitudinal study of about 300, if I recall correctly, nursing home residents in Japan. And they looked at what proportion of them became cognitively impaired over 6 years after baseline. And at baseline, they had characterized their dentitions. And you can see here that there is a bit of a gradient across these, categories, these ordinal categories of dental status, where the smallest proportion becoming cognitively impaired had more teeth at baseline. So there's some circumstantial evidence here, but it all depends on how you interpret it. And the authors of this paper themselves, in their trying to explain this association, pondered on the issue of whether tooth loss might lead to cognitive decline, but they were very, very cautious, rightly so. And they said that a great deal of further research is necessary. Well, we ended up with quite a lot of studies that climbed onto the bandwagon and even a couple of systematic reviews. And in fact, the findings of these 2 papers contradict each other, which just gives you an idea of how controversial the whole area is. And the mechanisms proposed by those authors. So the one that was first proposed was that if you have more missing teeth, you're not able to chew as well, so you're going to have a poorer diet. So you're not going to be as well nourished. That will ultimately compromise your cognitive function. Another one was if you've got missing teeth, you've got fewer intermaxillary contacts, so there's less proprioceptive feedback to the somatosensory cortex, and on a use it or lose it basis, maybe that compromises your cognitive function. That is very far-fetched, but this is what's been proposed. And then, of course, there's a periodontitis thing that the inflammation from the periodontitis has actually compromised the cognitive function. But of course, people are losing teeth because of the periodontitis as well. I recently, well, earlier this year, Yoram Barak, a colleague, a psychiatrist colleague, and I published this critical examination of this whole area, and I urge you to read it. But let's have a look. In this, we reproduce this diagram from a Finnish paper, and it's a very good one, looking at all of the influences on cognitive decline as we age. And there's a heck of a lot of them. And interestingly, in this paper, which was in the Lancet, the authors don't mention anything dental here at all, which probably should be a warning sign for us. If we go back to our pace of aging findings from the Dunedin study, this slide I showed you earlier, that these people here are aging much faster. We are seeing their cognitive decline by midlife. And, you know, this is a good 2 or 3 decades before they become old people. So we're already seeing the cognitive decline. So, you know, this really has to make us think. And the other thing to consider too is that there's a considerable degree of continuity in cognitive function. The people who have better cognitive function in childhood become the adults with better cognitive function. They also have better lives. They have better diets, they earn more, they live in better areas, they have better habits, they have better exposures as they go through life. All of that favors better cognitive function. They also have greater cognitive reserve later in life, so that when they do start to get cognitive decline, they've got more gas left in the tank before they run out of it, if you like. Now, this is data from the Dunedin study where we looked at childhood IQ. So here we have ordinal categories of childhood IQ. So here are the least intelligent children, and this is standardized childhood IQ measured on several occasions through childhood and then used to categorize these study members. So here are the most intelligent people. I want to draw your attention to the proportion missing 1 or more teeth. It's by age 38, this is 50%, 37%, 28%, 21%. Categorize it as 3 or more missing teeth. We've got 1/4, we've got 1 in 7, 1 in 12, and about 1 in 20. That's quite a gradient in tooth loss. So in fact, what is happening is not that the tooth loss is leading to the cognitive decline. It's the other way around. Their cognitive function early in life and through life is leading to them having more, leading to them losing more teeth as they're going through life. And if we have a look at the number of missing teeth here on average, you can see that gradient there too. So this is how it works. This is how we worked out that it works. So our cognitive function in childhood selects us into particular health and oral health trajectories. There's continuity in this through life. And the people with poorer cognitive function, by virtue of their life chances and their self-care and their exposures, lose more teeth along the way. And if we compare people with more teeth and fewer teeth at any stage along the life course, we will see an association with their cognitive function. It's not a causal one in the way that is being argued when people are interpreting these data from older people. It's not the tooth loss leading to the cognitive decline, rather it's the cognitive function leading to the tooth loss. And you know how we interpret data like that is very, very important. And we shouldn't either overinterpret our findings or misinterpret them. Here's an example of another one. My colleague Jonathan Broadbent went on a ship that did charity dental care in Papua New Guinea. And people in these villages were concerned about losing teeth because they thought it led to blindness, because they knew that there were higher rates of blindness and missing teeth among older people. Now, that's a ludicrous proposition, isn't it? It's no more ludicrous or less ludicrous than tooth loss leading to cognitive decline. How am I going for time, Coleman?
You're okay. We're just hitting time.
Right, okay. I was just going to say, just finally getting away from tooth loss and cognitive decline, it can be informative to make cross-cultural comparisons too. So we have done this with China and New Zealand, and we compared two key age groups, 35 to 44 and 65 to 74. And if you look at the two cultures, they're very, very different. New Zealand is a much more selfish, Western, it's all about me culture, whereas in China, there's a lot more reciprocity and mutual obligation and responsibility. You look after yourselves better. So these characteristics, these differences, we thought would manifest as differences in the oral health of these people by the time they got to old age. And that's certainly what we saw. If you have a look, here, just in the proportion dentate, the proportion with the functional dentition, the average number of teeth, and even in there were differences the other way in the periodontal disease and in terms of the periodontal bleeding. So there were differences in different directions depending on what you looked at. But these were quite fascinating. And again, I suggest that you read that paper where you'll have time to ponder those differences and what they mean. So when we think about interpreting oral epidemiological data, and particularly data on older people, number one, we've got to use our common sense. We've also got to think about socio-cultural influences, and we've got to think about how those people have got to old age and their journey through the life course. And we shouldn't think in a reductionist way. We've got to think in a socio-cultural, more of an epidemiological way, rather than treating people as biological units. So thank you very much for listening. I hope I haven't bored anybody too much. And it's been lovely to have the opportunity to talk to you. And I'd like to thank the IADR Southeast Asian Division for the kind invitation. And I'm also grateful to my co-researchers on the work that I've presented here. Thank you very much.
Thank you very much, Murray, for that really interesting talk, as usual, very thought-provoking. If I may go ahead with my presentation time, and if you can just confirm that you see that. Yes, all right, thank you. Our world is aging and aging fast. The pace of population aging is much faster than at any time in the past. The proportion and absolute number of older people is quite considerable at over 1 billion in 2019. Last year, for the first time, there are more people on the planet over the age of 60 than there are under the age of 6. Traditionally, it has been in high-income countries like Japan that we've had an aging population. But increasingly, and as will be the way in the future, there will be more and more older people living in low and middle-income countries, such as many of those in Southeast Asia. And this poses a considerable challenge to us in terms of health and social care systems. In addressing the issue of aging, the United Nations last year in December declared this decade to be the decade of healthy aging, with the World Health Organization being asked to implement 4 action areas. 1, change to be an age-friendly environment. The environment, physical, social, and economic environments are key detriments to our health and well-being, and it affects our mental and physical capacity across the life course, particularly so in older age as we adjust to loss of function. Another key action plan is combating ageism. Despite the massive contribution that older people have made to and continue to make to our world life, there is negative attitudes towards older people are common in the way we think, as in stereotyping, how we feel in terms of prejudice, and how we act in terms of discrimination, particularly in how healthcare is delivered, including oral healthcare. It is essential that older people have access to good quality integrated care, and that this care should include oral health, and that it not be a financial burden to older people. As older people age, significant proportions will experience significant declines in their capacity, both physical and mental, and as such, there's a limit to what older people can do for themselves, and they will require care from other people. And thus, likewise, it's important that they have access to good quality, long-term care, if required, to enable them to function and live with dignity. Important enablers for this are engagement of older people. Their voice is important to be heard and their autonomy respected. Leadership and capacity building, connecting stakeholders, and strengthening research data and innovation. With respect to the last enabler, strengthening research data and innovation, we've undertook a systematic review looking at the effects of oral health and successful aging. In other words, how is oral health operationalized with respect to successful aging? And registered this systematic review earlier with Prospero in 2021. Oratomo's model of successful aging, incorporating healthy aging, active aging, and productive aging, were used to guide identification of keywords for successful aging in the systematic review, relying on other systematic reviews in the medical literature with respect to healthy aging, active aging, and productive aging. The PRISMA framework guided us through the process. You can see over 1,000 articles, and eventually over 250 articles informed the review. Essentially what we were looking at is what are the common definitions, models, and concepts used to operationalize successful aging in the oral health context? What are the quantitative and qualitative methods and instruments used to assess it? And what are the cutoff levels that can be used to determine successful aging or not? Fernandez-Ballestor's framework for screening components of successful age dimensions and outcomes was employed to provide a qualitative synthesis of effective studies. The vast majority of studies considered biomedical models of successful aging, 4 out of 5 studies, 80%. And approximately a quarter considered psychological aspects, with very few considering the social dimensions. Most studies were unidimensional in their concept of what successful aging was, 88.9% of studies, or 241. 10% considered 2 dimensions, i.e. biomedical and another dimension, and only 1.1% considered all 3 dimensions. Within the biomedical model, the most frequent aspects considered were longevity or survival from mortality and nutrition. Within the psychological model, the most common attribute considered was cognitive function. As Murray referred to earlier, it's increasingly being looked at in the literature, particularly in recent years. Rarely, if ever, was social effects or social dimension to successful aging considered. And there are attributes relating to environmental mastery that we should look at further on. To look at these aspects, what I've chosen to look at is to say, looking at each of the main components, what evidence do we have to date in the literature? As aforementioned, a key outcome of successful aging has been survival. That is the longevity and length of your life. A recent systematic review published this year in the Journal of Dental Research looks at the effect of periodontal disease and its ultimate outcome, tooth loss and edentulous, and the risk of mortality. Edentulous was associated with 1.64 increased risk of all-cause mortality. Edentulous was associated with more than 3 times the risk of mortality from cerebrovascular causes of mortality and almost 3 times the risk from coronary heart disease mortality. It was also, edentulous was also twice the risk of cardiovascular disease mortality, and it was also associated with an increased risk of pneumonia and cancers. Looking locally in the region, we have a very interesting series of life course studies, if you will, on looking at the Chinese longitudinal health longevity survey, which has been running since 1998 with data collected every 3 years. I think the 8th wave of this, the data has just been released. And looking at the relationship here in China with tooth loss and the risk, relative risk of, or hazard risk of mortality over time, compared to those who are 20 teeth or more, those with 10 to 19 teeth at baseline were 1.14 times more likely to die in the analysis period over the 13 years. And those who had 1 to 9 teeth were 1.23 times more likely to have died in the intervening period compared to those with 20 or more teeth. And those who were edentulous, the hazard ratio was 1.35 times that of those who had 20 more teeth over the study period spanning 13 years. There was a nonlinear association between the number of natural teeth and mortality, with significant associations evident between tooth loss and mortality when the number of teeth retained was less than 25. Looking now at the effect of whether rehabilitation, or whether among that sample, whether they had lost teeth but they had some dentures, was this associated with a reduced risk of mortality? For dentures users, or those who claimed they had dentures, the risk mortality was lower. The hazard ratio was 0.81. Compared to edentulous and those who wore no dentures, those who were edentulous and wore dentures, the hazard ratio was reduced by 8%, or the HOR was 0.82. For those who had 1 to 9 teeth and did not wear dentures, compared to those with no teeth and no dentures, the hazard ratio was down 9% at 0.91. Wearing dentures, the hazard ratio was reduced 0.75, down 25%. For those with 10 to 19 teeth that wore dentures, the hazard ratio was down nearly 40% at 0.61. And for those who had 20 plus teeth, but at some aspect of rehabilitation, they had a 40% less likely in the hazard ratio compared to those who had no teeth and no dentures. There has been some systematic reviews that have looked at this concept about whether there is evidence to suggest that rehabilitation, even by the provision of dentures to those, removable dentures to those who have lost teeth, and whether that reduces the risk of mortality. Fewer edentulous individuals who wore complete dentures died over the follow-up period from this systematic review, with follow-up periods being anything from 3 to 24 years. Among the 5 studies that informed this review, 4 of the 5 studies did show a significant association between those who wore dentures and mortality rate compared to those who did not wear dentures and mortality rate. Of note, these studies vary considerably in size. I think one of the studies Murray talked about earlier, which was around 200 participants, to the largest Japanese study here of 5,000. And again, their age, the period of study ranged from 3 to 24 years. Of note, the one study who did not identify denture wearers as having a reduced prevalence of mortality over time was the smaller study with only 200 subjects. And actually, there was only 7 people in that study that wore dentures. The larger study, which involved over 5,000 subjects and involved a study of people over 4 years for those over age 65 and older in Japan, more than a third of those who had no conventional dentures died earlier compared to those who wore dentures. 16.3% died over the study period. The next important aspect to look at is in the biological was nutrition. This was very common. Older people are at risk for tooth loss, and this can compromise nutrition. Our review found it to be the most common area of biological health assessed with respect to active aging, and this is increasing over time. Oral health status affects food choice, and it affects food intake, as well as macro and micronutrient status, and ultimately this can lead to a rapid decline and sarcopenia, the loss of muscle mass. In assessment of nutritional status, the most common measure used is the mini nutritional assessment measure. This takes many forms. It's rather brief. It can be completed within 15 minutes, and the most widely used measure is an 18-question measure. A short form measure containing 6 questions can also be used for screening in identifying those at potential risk of malnutrition and those malnourished. An alternative measure is the Malnutrition Universal Screening Tool. This incorporates assessment of body mass index, weight loss, acute illness affecting food intake for more than 5 days to determine nutritional risks. Less often and rather brief, predominantly used just in hospital settings in the US, is the Malnutrition Screening Index, and this is used largely to assess recent weight loss and recent poor intake of food. A 2020 systematic review and a meta-analysis considered if tooth loss was associated with nutritional status. From 588 studies, 7 informed this review and 6 were amenable to quantitative analysis. Among the 7 studies, 5 studies identified a significant association between tooth loss and nutritional status. From quantitative analysis, meta-analysis identified that edentulous, or those who lacked functional dentition, had a 21% increased likelihood of being at risk of malnutrition or being malnourished compared to dentate or those who had a functional dentition. Controlling for other factors, including medical history, the relative risk of being at risk of malnutrition or undernourished was 1.32% likelihood for those who were edentulous compared to those who were dentate or had functional dentitions. Another earlier systematic review of 26 studies, of which 23 were cross-sectional in nature, I might add, found no significant association between edentulism and use of prosthesis with malnutrition status. However, quantitative analysis identified a significant association in that the standard mean difference in the number of teeth was 0.14 in subjects at risk of malnutrition, i.e. MNA scores 17 to 24, or those who were malnourished, MNA scores less than 17. This result while it may be statistically significant, it should be interpreted with caution, since its clinical significance may not be relevant, i.e. a difference of 0.1 teeth between those who were malnourished and not. Another systematic review which appeared this year looked at, in addition to it, longitudinal studies of it. They found no significant association. Sorry, my apologies. There's a growing interest in whether rehabilitation of teeth affect nutritional status. And this review from the Tokyo Institute of Gerodontology earlier this year looked at 4 studies comprising 3 randomized controlled trials, 2 of 6-month duration and 1 of 3-month duration. In all trials, the studies were the provision of complete dentures, and the intervention group received an additional intervention of dietary analysis. The outcomes were all positive in that those who received complete dentures and dietary advice had improved dietary intake, particularly that of proteins in their brief diet history questionnaire, and also an improvement in their nutritional association as reflected in M&A short form questionnaire. Another issue, although not widely seen in our systematic review, was frailty. And frailty in itself is a complex of symptoms characterized by a decline in functionality of different organs, with various ways to assess it, from phenotype to the broader deficit accumulation scores. Global estimates are that there's about between 1 in 20 to more than 1 in 2 people that are classified frail in the community. For the most part, the interest is associated with oral health features associated with frailty, with an acknowledgement that in part it may be bidirectional. There is also consideration of oral features of frailty within the mouth, and advances in its measurement has led to an increased assessment of this, which I'll refer to in a moment. Again, I'm going back to that local study in China of the Chinese Longitudinal Health Longevity Study. This is wave 7 of 2014, which involved 4,000 people. The frailty level was on average 27% among participants. The bivariate analysis showed an association between number of teeth retained and frailty. For those that had more than 20 teeth, the percentage deemed by phenotype frailty was 7.8%, and for those who were edentulous, it was approximately 44%. Looking at adjusting for factors which included sociodemographic factor, age, gender, education, economic resources and dependencies, both as a child and as an adult, behaviors of smoking, drinking, and exercise, and BMI, it was shown that for those with no natural teeth, there was an increased risk, almost a 5 times risk likelihood for being frail compared to those with 20 or more teeth. Last year, there was a very nice systematic review in Murray's Journal of Gerodontology, which looked at the different components of oral health that may contribute to this. This was comprised of 17 cross-sectional studies and 9 longitudinal studies. About the range of frailty ranged from 8% to 66%. A number of oral health parameters were considered, both clinical and self-reported. Among number of teeth, being edentulous or having less than 10 teeth increased the odds of being classified frail over the study period by approximately 2. Not using a prosthesis or being in need of a dental prosthesis was associated with also the increased odds of being frail, again, about 2. Untreated caries and severe periodontal disease was associated with approximately twice the likelihood of being classified as frail subsequently, and reduced oral function, things like and dry mouth, was associated with increased likelihood of being frail. Reduced oral function, we had something like 5 times the likelihood of being frail. Subjective oral health status like self-reports of pain, chewing disability, and global oral health were all associated with the risk of frailty up to 2 or 3-fold amount. A systematic review, again in gerodontology, focusing only on longitudinal studies, showed that all 5 studies identified association between oral health and frailty. 2 studies examined the association between number of teeth and frailty, and both found this to be significant. The first study, which looked at a period of over 3 years, found that there was for each tooth retained in the mouth, there was a reduction in frailty of about 5%. The other study found the 3-year incidence of frailty was twice as high for participants who were edentulous at baseline. Having a functional dentition was associated with a reduction of hazard risk of being failed by about 50%. And the other study, incorporating number of teeth as a frailty measure in itself, found that those who were classified as orally frail at baseline were more than twice as likely to be systemically frail later on. This turns to the point about the notion of oral frailty. Oral frailty is concerned with not just the number of teeth or the intactness of the dentition, but expands to include functional aspects such as chewing, swallowing, and talking. Much of this work has evolved in Japan, where it has become an important outcome of oral health policies. To date, there is limited research on the trajectory of oral function, and this in part relates to the requirement of training and having the measures to monitor it over time. Fortunately, recently, they have come up in the archives of gerodontology and geriatrics with an oral frailty index comprising a short form measure of 8 questions. And it has high sensitivity and specificity in cross-sectional physiological functional assessment, and also some evidence that it has a good longitudinal validity with respect to assessing frailty and disability over time. A 1-point increase in oral health frailty is associated with 1.3 times the likelihood of new onset of oral frailty and a 1.1 risk of disability. And a cutoff 4 seems to be the one with the highest sensitivity and specificity that can indicate new oral frailty occurring. The next issue is cognitive decline, which Murray looked at. And this, there's no doubt about it, that cognitive decline is a major contribution to disability and a cause for dependency in oral health. And to that end, there's obviously that based on theory and looking at Murray's recent journal, which really is a fascinating read, looking at the effects of tooth loss and dementia. However, as Murray's presentation has shown, there's little empirical evidence to show that missing teeth through these pathways actually lead to cognitive decline, but rather it's as a result of early childhood influences of cognitive function and those that occur in adulthood. And thus, as he rightly says, there is no evidence to suggest the use of rehabilitation is important in preventing the onset of cognitive decline. Nevertheless, it's important to look at some of the associations. Again, I go back to the Chinese longitudinal health longevity study, which here we're looking at 6 waves of the data over a 13-year period, following 8,000 over 60-year-olds and using the many mental status examination as a measure of cognitive decline. Over time, MMSE scores of cognitive decline decreased significantly. For every year, there was an equivalent decrease in MME scores of 0.12. But regardless of time, having more teeth was associated with better cognitive function. For every additional tooth, the MME scores was associated with an increase in their score of 0.9. Having adjusted for demographics, the score increased by was associated with an increased score of MME scores of 0.9. And in the final model, having adjusted for tooth number, the time or cohort effect, the socioeconomic status, underlying multiple health effects, and the time period, those, for every tooth retained, their MME scores increased by 0.1 about every year. In other words, participants with more teeth had a slower decline in cognitive ability. An interesting review to emerge, and that's interesting when I see Murray's paper of 2021 in Nature and Aging, is to say, when we're looking at cognitive function, we typically have relied on this memory scale, which is only a screening part. And this gives us very little indication as to how the actual cognition decline, what area of function is affected. And largely, essentially, there are 6 domains to consider. 1, learning and memory, that is recall and recognition. Language, as in terms of object naming, fluency, grammar and syntax. Perceptual motor function in terms of visual, reasoning and coordination. Executive function of planning, decision-making and responding to feedback. Complex attention, meaning sustained attention, divided attention and selective attention, and social cognition. From this review in terms of language and memory, there were 16 studies. Most were cross-sectional, 4 being longitudinal. Among these studies, which adjusted for other factors, being 12, a third identified that yes, oral health features of tooth loss, chewing ability, and periodontal health was associated with learning and memory and recall of words. In terms of perception and motor function, there was less studies, 7 studies, 4 which adjusted for factors, and 1 of the 4 studies identified that there was an association, and that association was with periodontal health status. In terms of language, specifically, there was 3 studies, 2 which adjusted for other factors, and 1 of which identified that tooth loss was associated with language skills. In terms of executive functioning, there were 13 studies, 9 which looked at association with other factors, and 5 which showed an association. 1 included this longitudinal study. Tooth loss, chewing ability, and periodontal health status was shown to be associated with cognitive function. In terms of complex attention, there was 8 studies. The majority of these controlled for other factors. And again, the majority, 6 out of 7, showed that oral health features of tooth loss and periodontal health was associated with cognitive function in the area of complex attention. In the area of social cognition, we didn't identify any particular study. Which moves me to the last point. Social dimensions appear to have been rarely studied with respect to oral health outside of the social or psychosocial impact in quality of life. 2 attributes seem important. 1 is social support networks, i.e., do people have care or assistance available? 2 hypotheses exist on that. 1 is having a direct effect all the time, and the other is the possibility that it has a buffering effect when we come to crisis. The other attribute is with respect to social capital, our sense of belongingness to that environment, and how we function. And just to finish on that, I want to show 1 interesting study that we examined in our systematic review, and that's a Japanese study of over 50,000 adults, and shows that some aspect of social capital at a community level, including civic participation, and at an individual level, hobby and sports activity at least once a week seems to be effect with respect to loss of tooth over the study period of 3 years, and roughly 8% had lost one or more teeth. Thank you very much for your time. Sorry if I've run a little bit over time with that. If I may Yes, if I may now ask Dr. Varuna Pitfat to share her presentation and some findings from oral health and future directions in Thailand. Thank you very much.
Thank you, Coleman. Can you see my slide now?
Yes, we can.
Okay, great. All right. Hello, everyone. It is my great pleasure to be here today with you to share our experience in Thailand as to how we integrate oral health into national healthcare policy for aging society. And I would like to thank the organizing committee, especially Professor May Wong, for inviting me here. After listening to a very amazing research from New Zealand, from Murray, and a comprehensive review of oral health and aging, healthy aging, we must wonder what could we do in the real world. And I would like to share our experience. And here is my outline. I will start with the current situation of aging population in Thailand, and then share how we integrate oral health care in the national healthy aging scheme and national oral health plan for older adults in Thailand. Population aging is a major achievement of development, as it is a result of both increased life expectancy at birth and the reduction of total fertility. And Asia seems to be doing very well about this. Asia is the fastest aging society in the world, and Thailand's aging society is just behind Japan, followed by South Korea and Singapore, and we are just slightly ahead of Vietnam. In fact, Thailand is becoming an aged society next year, with over 14% of population aged 65 and above. And we are actually, sadly enough, we are the first developing countries in the world to become aged society. So that means we already aged, but we don't have much money as compared to others. Here is the graph showing the percentage of population over 65 years. And you can see from these lines of Japan and Thailand, the slope of Thailand, it's very much the same as Japan 30 years ago. So we follow the patterns to be aged society, but we are not as rich as Japan. So what can we do in this situation? How could we prepare for this socially, economically, and also how we do with the health and wellness of the people? If you look at the oral health status of population age 60 to 74 years, this is the indicator of WHO, the percentage of adults with over 20 functional teeth. This data is from Thailand's National Oral Health Survey, which was conducted every 5 years, and we use these data mainly to monitor oral health status of our population, as well as to evaluate the effectiveness of public health care system in the country. And you can see here that the percentage of people with 20 plus functional teeth increased a little bit over the years, from 1984 to 2017. So it's increased only about 10%. The prevalence of dental caries is very high in this population. Almost everyone have some form of dental caries with the DMFT of almost 16. And most of these carie affected teeth are missing. So the mean number of teeth at this age is about 18. When we look at the percentage of edentulisms, the percentage reduced, which is improved from 16% in 1984 to about 9% in 2017. So it seems we still need to do a lot more to improve the situation. Before I talk about the oral care policies, let me explain a little bit about healthcare system in Thailand. The country has 76 provinces, which could divide to districts, sub-districts, and then village. And in each village, we have village health volunteers, which is primary healthcare workers, who is the backbone of the public health system in Thailand. For the family, right now we start the system of family health volunteers as well, but it's still very small. From primary health care, these village health volunteers would contact with health center, sub-district health centers, and then going up in the system, it's community hospital and regional hospital and university hospital in tertiary care. We have a very good referral system throughout the country. Thailand is one of the countries that enjoy the benefits of oral health universal coverage. Actually, more than 99% of the population right now is covered by some form of public health insurance. And most of the insurance is the universal care coverage, which is about 3/3. Other form of insurance is, for example, social security system and the civil service government system. We have a life course approach involving prevention and promotion, and also cover the treatment for pregnant women, for early childhood, up until school age and adolescence and working age and elderly. For the elderly adults, we have Variwanage as the main prevention and promotion program, which is covered by the universal care coverage. And for the treatment side, it covers filling, scaling, extraction, pulse treatments, and acrylic dental prosthesis. So this is free of charge for everyone. So in order to work on geriatric care for older adults, we work hand in hand with other health professionals. We put mouth into the body by providing comprehensive healthcare for older population, with the goal to have healthy, independent, and having good quality of life. Our goal for the oral health part is for the people to have at least 20 functional teeth, so that they will be able to chew, swallow, and have a confident smile. The care is provided like holistic care. We start with the prevention system in the people who are at risk. We focus on screening and the disease prevention among those at risk. And among those who are healthy, we try to delay degeneration of the whole body. We promote elderly clubs and school system for adults so that they have social participation. And for those who need rehabilitation, we provide free acute care. And for dependent living, we have the system to promote caregiver. We have the budget allocated to the local authorities so that we can support caregivers for those dependents living at home or home care. For oral health care in aging population, we focus very much on oral health promotion, increased knowledge, and proactive health prevention and promotion, as well as self-care. And we give this oral health promotion package to all the segments, healthy, homebound, and bedbound. For healthy persons, we have health promotion activities, especially through the elderly club. And for those with homebound and bedbound, we have village health volunteers and caregivers who provide the primary care before going or contact to the higher level of care. Thailand has a 20-year national strategies, which also include public health strategy plan. These plans have 15 urgent flagship projects in public health, and Thailand's national oral health plan for geriatric populations is one of them. Actually, this plan started in 2014. At that time, geriatric dentistry is a very new field in the country. We have only 1 or 2 people who study in this field. Most people are specialties such as periodontology or prosthodontics, but just a few that learn about giving comprehensive care for older adults. So the plan started up with 4 strategies. The first one is to develop the model oral care system for older adults in the public system through the Ministry of Public Health. Second strategy is to develop research technology and innovation for this population. And the next one is human resources and dental personnel development. As I mentioned, at the time, there are very few people study geriatric dentistry, but then the government would like to develop the oral care system specifically for this population. So there's a really need for us to get some training. So the government provide scholarships for those in dental schools to study abroad or take some short courses so that we learn from Japan or Canada or other countries in Asia and the US so that we come back and can share our knowledge to people to dentists and dental nurses in the public health system. So right now, almost all the dental schools have geriatric dentistry curriculum that could provide short course training or comprehensive training for dentists in the Ministry of Public Health. Strategy 4 is for the management and support, produce the monitoring system and evaluation of the overall projects. And we have the aim to increase the proportion of prevention and promotion to treatment and rehabilitation for older adults, which initially was 20/80 to be 80/20. And the overall goals of the National Oral Health Plan is to have 80% of these elderly to have good oral health and at least 20 teeth, functional teeth. And also have some innovations to solve oral health problems in the elderly. And here is the indicators of the National Oral Health Plan. We have leading indicator and also lagging indicator, and the main plan is for 20 years. So our main goal is to have 80-year-old individuals to have at least 20 teeth remaining. So at the end of the 20 years, we hope that this percentage would be 80 among 60 to 74-year-old adults and 50% for 80 to 85 years old. So the key activities is we have the participation of public sector, the dental school, Ministry of Public Health, and all those in medical and health sectors coming together to increase the access to care. We have the collaboration with the long-term care facilities as well. And we focus very much on village health volunteers and caregivers who can contact with the elderly firsthand. So these people would connect between the individuals and the health sectors, the dental nurse in the sub-district and dentists in the provincial hospital. And at the first phase of the project, we have the goals to have 60% of the elderly to have good oral health and proper functional teeth, and having 3 comprehensive innovation to promote. And the result is that we are able to establish the oral health promotion system for the elderly, starting from hospital base, which could provide comprehensive care. And in the community base, we focus on the oral health promotion in the elderly club, integrated with various partners, including long-term care activities in sub-district health promotion hospital, health promoting temple, and training course for caregivers. And we have the system to provide the dentures for the elderly. This is to promote functional teeth. So our goal to have people to be able to chew the food and have proper nutrition. So we have denture delivery, and also we have dental implant placement as the implant written denture. These implants are Thai developed, so it's local made and implemented and tested to be used in the public sector in Thailand. We also have the oral screening application for pre-malignant disorder screening, which can be used by village health volunteers who are trained and can refer the patients to the health sector. Also, we integrate oral conditions with the NCD care. We have the NCD Clinic Plus guidelines so that we can integrate oral health assessment and care within the NCD clinic. This is a pilot project in 35 provinces throughout the country. And for the second phase, which will end next year, we will focus on the self-care and increase access to care with the participation of health service from community to private sector. So during this phase, we will integrate self-care and increase access to care as well as innovation. We have created screening tools to be used by the older adults and also for health volunteer, so that they can have a basic screening and communicate to the sub-district hospital. We also created the management guidelines, integrating NCDs, all of oral health and NCD together. These are web-based resources, and so by this, we hope that we could improve our data system as well. This is the example. This is Blue Book application. In Thailand, for children, we have Pink Book application to monitor growth and development of the children. And the Blue Book is just developed to do the same things for the elderly, so that we can do geriatric syndrome screening, and some oral health screening are included as well. And this will be used by the adults, the elderly themselves, and also can be used by the village health volunteers. So on both sides, they can enter the data, they can verify the data, and once anything happens with the older adults, the village health volunteer will connect with the sub-district officers and the community hospitals, so that the elderly could get proper care. So we also have the improvement of data system. So we have data system in the health center, in the long-term care facilities, and we have right now the real-time data that we can see from those screening tools. And these still need some validation and checking of the validity of the data. And next year will be the last year for the National Oral Health Care Plan for geriatrics. And what could we do next? During the time, we already integrate oral health with the healthcare plan for elderly. So this will be healthcare provision, what we call elderly seamless care, integrating oral health with other aspects of health for the elderly. And we will move from elderly persons to pre-aging. So we will move more on the working group, the pre-aging group, and focus on promotion, prevention, as well as consumer protection. And we focus on screening, community-based screening, and the management, the referral system improvement, so that the persons could have proper care immediately. Then at all level, we will focus on the increase of health literacy. So way forward, we will emphasize on promotion, prevention, and protection programs. We'll expand oral health service system, and because right now, like 50% of dentists in the system working in public and 50% in private. In order to increase access to care for the patients, we now thinking about adding the private partner into the reimbursement system for the insurance system. And also, in order to reduce disparity in oral health care, we try to adjust the three main financial schemes, which is at the moment, there are some differences among the schemes. So we try to make it, adjust it to be as similar as possible among the working age group, so that when they can receive proper care. And at the beginning, when we start the National Oral Health Plan for geriatric population, we have very little research on the elderly. So when we formulate or establish the plan or the program, we use data from other countries. Very few data are from Thailand. So right now we try to increase local research so that we can have appropriate research for the countries. And also we need to do program evaluation of what we have done so far. So if I map what we have done in Thailand with the World Health Organization global strategy on oral health, mapping is very good actually in all principles. We use a public health approach. We integrate oral health in primary health care. We use the new oral health workforce model to respond to population needs, which is the oral health village health volunteers. We have people-centered oral health care. We tailor oral health across the life course and also optimize digital technology for oral health. So I would like to thank all these people who helped me with the presentation. I got a lot of slides from Dr. Pieda Prasad. I would like to thank all of these. And I'll end my slide here. For the situation of Thailand, actually, we don't have much evidence from research when we start a project. But then with the pressing need to have the system providing for our aging population, we need to do something. And then we have this oral health care plan, which is not perfect, but we have things well in place to prepare for these population in the future. Thank you very much.
Thank you very much, Dr. Wanak. I think it's really interesting. I haven't seen such a comprehensive plan in any other country. So really well done. I just love the integration of the model. Just really good. And thank you for sharing that. We are fairly tight for time, but if we have any questions for any of the panel members, I'm sure they'll be happy to answer. If not, we've just got a note from Professor Wong from May to remind the delegates that we will have the award ceremony at 4:30 in the virtual oral session hall number 1. Many thanks again, Murray and Warunak. Really great to hear, and at least we're all going on the same theme. It is interesting that this is our aging population. This is the decade for change. So let's work towards that and the evidence. Thank you very much. Cheers.
Thank you.
Thank you.
Thanks. And with that, I should close it. May, do you want to say a few words?
Thank you very much for all 3 excellent speakers. It's really a different perspective, but very comprehensive in bringing this oral health and successful aging together, which is something really emerging, and we have to deal with it instead of like just wait and see what will happen. So we really see different levels from the longitudinal studies, from the review, all the reviews, and together with the policy. It will be good that we can put all these things together and then really to take the challenge, how we tackle the problem, or I shouldn't say problem, how we can really move on with successful aging, so that everyone will really enjoy the time, not just thinking about aging as something bad, but really that we can still do it successfully. So thank you again so much for all the 3 speakers. And actually this concludes our symposium, but at the same time also the program. So because the other oral sessions, they are also in parallel and should be finishing by now. And now it gives our judges a bit of time to work out all the judging scores, and then we'll come back with the award ceremony at 4:30 and closing remarks. Thank you very much.
Thank you.
Thank you.
Thank you.
Thank you.
Cheers. Bye-bye.
This would normally be the time when we go for a drink. But
Yes.
Yeah. I look forward we can meet in person and have a drink.
sometime.
Next time.
Soon. Okay. Bye-bye.
Bye-bye.
Bye.